Hello everyone, today we're gonna present you our project, the effect of crude Mannoprotein extract from Saccharomyces cerevisiae cell wall on the regeneration of Dugesia japonica for the development of a wound healing cream, presenting by me Kankanit Walaor, Bashiravit Panpet, and Apiwit Thongtae. And also thank you to our advisor, Dr. Arawan Piyabun.
Wounds are common conditions that can occur to anyone. If not treated them properly and promptly, they may lead to inflammation and increase the risk of the microbial infection. In general, wound treatment involves the combined use of povidone iodine and antibiotics. However, povidone iodine may impair wound healing, as most studies report that povidone iodine delay healing or increase the infection rate. The next study found that nearly half of the tested antibiotics accelerate healing, while some had no effect or even delay healing. Therefore, our project focuses on a bioactive substance that can inhibit microbial growth, reduce inflammation, and promote wound healing, namely Mannoprotein. Mannoprotein is a component of the cell wall of the yeast species Saccharomyces cerevisiae. Mannoprotein is composed of mannose sugar linked to proteins, as shown in the middle of the presentation. Based on the literature review, Mannoprotein has the following properties which are very beneficial for wound treatment. Due to ethical and safety limitations in human testing, our project used the Panarian Dugesia japonica, a highly regenerative and convenient model organism, to study wound healing. After the test, the yeast extract was incorporated into a cream formulation. Overall, our experimental procedures include 5 steps as follows.
First, culturing Saccharomyces cerevisiae or yeast. We're gonna culture yeast on YPD agar before transferring them into YPD broth and incubating them. Next, we're gonna extract Mannoprotein crude from yeast. The process of extracting Mannoprotein crude is quite complex. It involves several techniques and instruments. For example, centrifugation, evaporation by a rotary evaporator, and also freeze drying. After completing the processes, we obtained crude Mannoprotein extracts. We performed the extraction twice, and we got the percentage yield of the extraction was approximately 18 to 19% in both experiments.
Next is the chemical composition analysis of our extract. First, we determine the total carbohydrate content using the phenol sulfuric acid assay. We construct a standard curve using the glucose as the reference standard and measure the absorbance at 490 nanometers. Next, we determine the total protein content using the Lowry assay. A standard curve was prepared using BSA or bovine serum albumin as the reference standard, and the absorbance was measured at 750 nanometers. As a result, we construct a glucose standard curve. The graph shows a strong linear relationship with an R-squared value of 0.99 and the linear equation below. Using the linear equation, we were able to calculate the carbohydrate content in our extract. The crude extract from 65 microgram per milliliter of yeast contains 24.3 microgram per milliliter of carbohydrates, accounting for 37% of the total extract. And then we construct a BSA standard curve for protein analysis. The graph shows a strong linear relationship with an R-squared value of 0.99 and the linear equation below. Using the linear equation, we were able to calculate the protein content in our extract. The crude extract from 65 microgram per milliliter of yeast contained 8.247 microgram per milliliter of protein, accounting for 13% of the total extract. To evaluate the antimicrobial activity, we used the method called disk diffusion method. And the experiment was conducted in 2 concentration ranges, 50 to 150 micrograms per milliliter and 100,000 to 300,000 micrograms per milliliter. At 50 to 150 micrograms per milliliter, our extract was unable to inhibit bacterial growth. However, at 100,000 to 300,000 micrograms per milliliter or 10 to 30% weight by volume, the extract showed inhibitory activity against the bacteria.
Next, let's move to the methodology of parallel regeneration test, where we add a crude extract solution from yeast cell wall concentration 50, 100, and 150 microgram per ml into the well plate, and then we cut parallel into the T part, take photo, and soak parallel part into the various concentration for 15 minutes. Once the 15 minutes are up, we transfer the parallel piece into the drinking water, and we observe and photograph the parallel daily under the stereo microscope for 5 days. Then we use ImageJ program to measure plasma to initial area ratio and plot the result. This is the parallel result. Left one is the initial area after cutting, and right one is a regenerate plasma. And equation plasma ratio on day n equal to the plasma area on day n divided by initial area. This is the parallel result. If you look closely to the left one, head part show the careless and well-differentiated growth trend, marked with a star. And on the other hand, body and tail show a growth trend is closely overlapped with a control group. This indicates that this extract has more visible boosting effect on the head piece because head have the central nervous system, lead to a higher regeneration potential. And this is a cream development. Cream formulation, there is a three main ingredient: oil fat, aqueous fat, and crude extract. To prepare, we heat oil fat and aqueous fat separately and combine at 75 degrees Celsius. And this is a physical evaluation of cream. First is a pH test, and second is spreadability test. In pH test, we dilute cream with a water 1:10 ratio and allow it to settle and measure supernatant with pH meter. And spreadability test, we put 0.5 gram of cream on the gas light over the graph paper, then cover it with another gas light and apply weight 50, 100, and 150 respectively. Measure spread area of the cream. And this is a pH test result. The result indicates that this extract did not affect change in pH of the cream. And this is a spreadability test, 50 to 100 gram. There is no significant difference in spreading area, and in 150 microgram per ml cream show a significant smaller spreading area. This indicates that higher extract concentration increased cream viscosity.
To conclude, our project can be summarized into 5 main findings. First, the crude extract is composed of 37% carbohydrate and 13% proteins. Second, at high concentration, our extract can inhibit the growth of Staphylococcus aureus. Third, the cream formulation maintained a stable pH between 6.76 and 6.81, which is compatible with wild environment. Fourth, incorporating the extract at 150 micrograms per milliliter increased the cream viscosity, resulting in a smaller spreading area. And fifth, the crude extract demonstrates a positive trend in promoting Dugesia japonica tissue regeneration, particularly in the head section. And that's all for our presentation. We hope you found our project interesting. Thank you for your attention.